In this lecture, in Chapter 4, we are going to talk about cells. Now, if this was a face-to-face course, this is usually where I either hear groaning, there probably is internal groaning, I might get a few eye rolls, because you hear and talk about cells in so many science courses. Well, it's true, every living thing is made up of cells, so you gotta know the basics. Now, there are two basic types of cells. There are eukaryotic cells and there are prokaryotic cells. We, humans, and anything in the animal kingdom have eukaryotic cells. And so in A&P, in biology courses, in most of your science courses, you talk specifically about eukaryotic cells. That's what we're made up of. In this class, we're talking about prokaryotic cells. The cells that we are going to be discussing this semester are all very tiny. It's microbiology. They're very tiny cells, and most of them are very simple. Bacteria don't have a brain and body parts. They are very simple organisms, and they're made up of very simple cells. So there are some differences between the two types of cells. So in prior courses, you've talked about eukaryotic cells. In this course, we're going to talk about prokaryotic cells. And so in this chapter, we're really going to be looking at what are the differences between them and what are some unique things that prokaryotic cells have that eukaryotic cells don't. So one big difference between a eukaryotic and a prokaryotic cells, other than that, eukaryotic cells are very simple, is they don't have a nucleus. They don't have membrane-bound organelles, meaning they don't have organelles that have a phospholipid bilayer. That is just way too advanced for them. An example would be mitochondria. It's something that has a phospholipid bilayer on the outside of that organelle. It even has two phospholipid bilayers. That is just way too advanced of an organelle. So they're missing all of those advanced structured organelles, and they're also missing a nucleus. Every living organism has these basic characteristics of life. These are the things that make living organisms alive. They can do reproduction, they can do growth, they can do some type of metabolism, they can have some type of movement or irritability, allowing them to go towards something they want, away from something they don't like. They can react, and they're going to have some type of cell support structures. They can also do a little bit as well of transport of nutrients and waste in and out of cells. Why I talk about these characteristics of life is because you're like, well, yeah, because we're talking about living organisms. But are we talking about living organisms in this entire class? There is a whole group of organisms that we talk about this semester, the viruses. Viruses don't have all of these characteristics of life, so they're not technically considered living organisms. We talk about them as they're all living organisms, but they don't meet the basic characteristics of life. They can't do reproduction and heredity on their own. And I'm like, they have to hijack cells to do some of these different things. So most of the organisms we talk about this semester are living. However, viruses still meet these basic characteristics. A bacteria, that we are going to talk a lot about bacteria this semester, so we're going to talk about the basic structures of bacteria cells. And bacteria cells, they are prokaryotic. They are simple cells. You're like, well, that doesn't look all that simple. But you don't see the mitochondria listed in here. You don't see those endoplasmic reticulums. I'm like, you're missing a lot of organelles. They are simplified structures. They're simplified cells. Now we're going to start looking at some of the outside structures and work our way in. Now some bacteria have different appendages found on the outside. They also have or can have what's known as a glycocalyx outside structure. So we're going to start talking about some of the appendages that some bacteria have. They don't all have these specific structures. A couple outside appendages is the ones that are used for motility, the flagella, and ones that are used for attachment, the Fimbriae and the Pili. So the flagella, you're like, well, we've heard of flagella. They're in eukaryotic cells. They are, and their function is the exact same. They are both used for movement. However, internal structure is slightly different between a eukaryotic flagella and a prokaryotic, and how they work, even though they work for movement, but how they work is slightly different. A prokaryotic flagella rotates in a 360-degree circle. And so I'm like, they kind of spin around. They move kind of like a boat propeller moves the cell like it moves the boat. And I'm like, and it gets that cell to move. But eukaryotic flagella, they move more in a whip-like fashion. They don't spin around like a propeller. Different bacteria do have different arrangements of flagella, and so if a bacteria needs to move, they have to coordinate some of these flagella. Again, these are simple celled organisms, and so their coordination isn't always that great. As I said before, I'm like, not all bacteria do have a flagella, but if they do, they can even be arranged differently, and it's species specific. So underneath the microscope, if we see flagella, we can look to see, well, where are they? How many are they? And it can help us identify what type of bacteria it is. And so some are just going to have a single flagella at the end. Some have like a little tuft or a little bunch of flagella at one end. Some have flagella on each side, on the opposite ends, and some are completely covered in flagella. Now, those flagella, when they're attracted to something, when the bacteria is attracted to something, whether it's some type of chemical, some type of light stimuli, and I'm like, or if they're repelled by it, those flagella can help the bacteria move. But the flagella just move in that circular rotating fashion. And again, they're not very coordinated. And so depending on how they rotate, they kind of move around in what are called as runs and tumbles. And so if the flagella are not all that coordinated and all of these flagella are spinning around all at once, the bacteria will look like it's just spinning in circles. It doesn't go very far. But then all of a sudden, half a second later, they start to coordinate. They all work together and they will shoot that bacteria forward in what's known as a run. And then it kind of gets back into that uncoordinated state a few seconds later and it does a tumble. When they're kind of just moving around, they're not really attracted to anything, they still are using the flagella. The flagella are moving and they kind of just zigzag all around. If they are attracted to something, so they want to get from the left side of the screen to the right, they can do it, but again, it's not the shortest distance here. They do all of these runs and tumbles and eventually make their way to where they need to go. Now, some bacteria also have a type of flagella called periplasmic, that it doesn't extend out, instead it's almost attached to it, and it causes a constriction of the bacteria. So the bacteria almost look like they're inching along like a little inchworm, and it can allow the bacteria to still move and get to where it needs to go. Those are a little more rare and very hard to see underneath a microscope. Some of the bacteria have the Fimbriae. It's still an outside structure. They are not for movement. This is for attachment only. Looks-wise, they kind of look like the cilia in the appearance, not function, but in appearance. They're very short. They're just another extension that can happen and allows the bacteria to stick to each other, because these are little sticky little hairs that stick out, and it allows them to stick to surfaces. So they can stick to each other and they can stick to surfaces. Another attachment is called a pili. These are more of a tube-shaped structure, and anytime bacteria connect together, if they have or can make this pili, when they're connected, it's called a conjugation pili. Now, it's used so that they can connect to other bacteria. It does not have to even be the same type of bacteria. And once they're connected through that tube, through that pili, they can actually exchange genetic information. It allows them to talk to each other. For bacteria, that's great. If bacteria are talking to each other, exchanging information, if one bacteria has figured out how to be resistant to a certain type of antibiotic, it can tell another bacteria how to do that by sharing DNA, sharing genes that code for resistance to certain antibiotics. Anytime bacteria talk, that's good for them. But if it's good for them, it's always bad for us. This is one of the top ways that antibiotic resistance is happening, is that once one bacteria has figured out, once it has the genetics and how to be resistant to antibiotics, it shares that information with other bacteria. And again, it doesn't even have to be the same type of bacteria. Another outside structure that they can have is a Glycocalyx. This is attached to the outside, and there are two types of glycocalyces. One is called a slime layer, and you can see down below here that yes, it kind of looks a little slimy. It's loosely attached. It is more water soluble, and usually the bacteria that are found in more moist environments would have a slime layer. And they can also have a capsule, which it's a lot more organized. It's this outside structure here. Now, a slime layer, they both can allow and help the bacteria to stick to surfaces, but the capsule has a few additional features to it, or a few other functions to it. They both can help a little bit with dehydration and nutrient loss, but the capsule is going to be better at it. And the capsule really helps inhibit our white blood cells from eating the bacteria. Again, the whole goal of a bacteria is to survive and hopefully thrive. And a capsule helps it do that. Our white blood cells can recognize bacteria as foreign invaders. It can find them, eat them up through phagocytosis, and get rid of them. These capsules are made up of sugars and proteins. And that in itself, a lot of our cells have sugars and proteins on the outside of it. And so right away, our white blood cells might not recognize it as foreign. So it kind of becomes a little bit of camouflage for our immune system that helps the bacteria survive. Good for it, bad for us. It also can make it a little slippery for our white blood cells. So our white blood cell, again, wants to eat these foreign invaders, but sometimes when it tries through phagocytosis to kind of grab around that bacteria, it's a little too slippery and it can't get fully around it to eat it. Again, great for the bacteria, bad for us. When we look at some of these underneath the microscope, we can actually see the capsules themselves. We can do a specific type of stain called a capsule stain. The capsule, all the white part, doesn't pick up the stain. And so it can show us which bacteria have capsules, because not all of them do. And so through a certain staining process, it can also narrow down, this is a bacteria that has a capsule, and that can narrow down what bacteria it might possibly be. Either of the glycocalyces, whether it's a slime layer or a capsule, can also aid in creating what's known as a biofilm. The word means a living covering. So it helps the bacteria stick to each other and stick to surfaces and completely cover surfaces. This could be completely covering the surface of various body parts. You have biofilms in your body. You have a lot of biofilms in your mouth, and especially on your teeth. We can also find biofilms on medical equipment. And it becomes a big issue is because once they all hang out together in these biofilms and coverings, when they hang out together, there is strength in numbers. And so they're a little bit harder to kill, and they could cause disease. This is showing an example of some of the bacteria that are on the catheter. And so these bacteria are sticking to each other, and they're actually sticking to the catheter itself. This happens more commonly with long-term catheter use, and it's one of the main reasons why catheters need to get changed regularly, because the longer that they are in, the more likely and more opportunity these bacteria have to stick to each other and stick inside those catheters and cover them and cause different types of disease. Now, another outside structure is a cell envelope, which is made up of the cell wall and the cell membrane. It's just the outside part of the cell. All prokaryotic cells have a cell envelope. They have cell walls, they have cell membranes, they've got this outside envelope part. Not all bacteria have the capsules, not all bacteria have pili, not all bacteria have flagella or Fimbriae, but all bacteria are going to have a cell membrane, and I'd say 99.9% of them all have a cell wall. It helps maintain that outside cell integrity, that structure. The cell wall part of the cell envelope, there are different thicknesses in cell walls. Bacteria, they're going to have a very thick cell wall, or they're going to have a thin cell wall. Now, the cell wall itself is made up of a structure called peptidoglycan. It's a big word, I use it a lot. Peptidoglycan makes up the cell wall. Other plants out there, they have cell walls not made up of peptidoglycan. This is specific for bacteria and for prokaryotic cells. About half of all the bacteria out there have a thick cell wall, and we call them gram-positive bacteria when they have that thick peptidoglycan layer. And about half of all the bacteria out there have a very thin cell wall, that thin peptidoglycan layer, we call them gram-negative. The interesting part about gram-negative bacteria is they have two cell membranes. They have their normal inner cell membrane, and they have a bonus extra outer cell membrane. So they have two cell membranes. Now the cell walls, they're made up, that peptidoglycan is made up of these repeating units called NAMs and NAGs. Now, they stick to each other, top down, side by side. It's very much like stacking up bricks on a wall. They're attached to each other, top down, side to side. They're all going to have some type of glue or something to adhere them all together. And when they're put together well, they make a very strong, very durable cell wall. Now that cell wall's function determines the bacteria's cell shape, and it prevents lysis. It prevents that bacteria from breaking apart due to changing water pressures. Now, that gram-positive cell wall, the big thing you need to know, you need to know this, star it, circle it, write it in big bold letters somewhere. Gram-positive bacteria have that thick cell wall. And we're going to talk a little bit coming up and a lot in the future, lab and in some lectures, that when we gram stain them in a lab, is that they do appear purple after a gram stain. Because again, about half of all bacteria out there have this thick peptidoglycan layer. We call them gram-positive. It means once I do this gram stain in a lab, if I can narrow it down, saying it has a thick cell wall or a thin, I've just narrowed out or eliminated half of all the bacteria choices out there. And there are antibiotics that work specific for gram-positive bacteria, and there are antibiotics that work specific for gram-negative bacteria. So without even IDing the bacteria, knowing exactly what bacteria it is, just knowing that it's a gram-positive, you can start to treat with antibiotics. In a gram-positive cell wall, they do also have another structure called a teichoic acid or a lipoteichoic acid. And the teichoic acid really helps, here's that thick cell wall, really helps anchor that cell wall to the cell membrane that's below it. So it helps maintain a little bit with that, the maintenance of the cell wall. It can help the cell wall move around a little bit. It sometimes can also stimulate a specific immune response because our immune system can sometimes recognize the teichoic acids if they're sticking on the outside. Gram-negative bacteria, the ones that have that very thin cell wall, that thin peptidoglycan, they appear pink after a gram stain. And again, they have that bonus extra outer membrane. Now, why I care so much about that bonus extra outer membrane is because in that membrane, they have something called a lipopolysaccharide. It's a lipid, it's a sugar structure altogether, and it's found only in that outer cell membrane. And why we care so much about it is that bacteria that have this lipopolysaccharide in it, that lipid portion of it, the lipo of the lipopolysaccharide, is an endotoxin, which means when that bacteria gets broken down, so when our immune system recognizes the bacteria, it eats it, it breaks it down, that endotoxin becomes released. And that endotoxin can cause a whole bunch of different types of responses in the body, none of them good. It can trigger fever, it can trigger inflammation, it can trigger shock, it can trigger blood clotting. It's why gram-negative bacteria usually have more symptoms than gram-positive bacteria. So if someone has a gram-negative bacteria infection, they usually are going to have worse symptoms just because as the body is starting to kill them and break them down, and I'm like, it's going to release that endotoxin. So they cause a lot of issues, is those gram-negative bacteria. So this is just showing a comparison between the gram-positive and gram-negative. Again, the thickness of the cell wall. Gram-positive has a very thick cell wall. Gram-negative, very thin cell wall, but a bonus extra outer membrane. Now the gram stain that shows us whether it's purple or pink in a lab, so that we'll then know if it's a gram-positive or gram-negative. We are going to talk about this later in lecture because it is so important, and some of the labs will also go over the gram stain. I'm just going to go over the really quick difference, and it all has to do with the thickness of the cell wall. We use two different colors in a gram stain. We use crystal violet, it's purple, and we use a stain called safranin. They say it's red, like technically it's red, but it shows up as pink when you look at it under the microscope. Well, gram-positive and the thick cell wall, that's, you're going to add crystal violet purple first. And both of the bacteria, their cell walls are going to pick up purple. However, gram-positive has such a thick cell wall that it holds on to that purple because it's got so much of it in it. It holds on to that purple throughout the whole staining process. And so when you get to the end, it still looks purple. Gram-negative has such a thin layer of cell wall that it will pick up purple, but because there's not much purple that it holds on to, it loses the purple right away during the decolorization step. And so then it has no color because it loses the purple, and it will pick up the second color that we add, which is pink, the safranin. And so underneath the microscope, if you see something purple, you have a gram-positive bacteria, and you've narrowed down it to, half of the bacteria choices. And if you see something pink, you've got a gram-negative, and you've narrowed it down to one of the gram-negative bacteria that are out there. Now some non-typical cell walls. So again, we talk about gram-positive, gram-negative, but there are some non-typical. There are bacteria that are in the genre of Mycobacterium and Nocardia that can make kind of a cool structure. They can make something called mycolic acid in their cell walls. Now, mycolic acid is a lipid. Yes, it's an acid, but it's a lipid. And when we gram stain bacteria, if we suspect a Mycobacterium or a Nocardia, and we tried to gram stain it, it wouldn't gram stain accurately. And that's because the stains we use, that crystal violet and safranin, are water-based stains. And if a bacteria has this mycolic acid, lipid, in it, it's not going to stain correctly. You can't stain a lipid with a water-based stain. And so in that case, if we suspect a patient has a bacteria in one of these two genera, we would have to do a stain called an acid-fast stain that is specific and will specifically stain that mycolic acid a certain color. And in lab, we're going to go over those acid-fast stains. And there are a few bacteria that don't even have a cell wall. They just don't have it. One of the big genus that doesn't have it is Mycoplasma. Now, because the bacteria gives the cell wall its cell shape, we call these bacteria, their cell shape is what's known as pleomorphic. It means they can change their shape. They don't have a specific shape because they don't have cell wall maintaining that specific shape. Now, bacteria do still have a cell membrane, and just like eukaryotic cell walls, it looks the same, and it does most of the same. It's a phospholipid bilayer. This should look exactly like what you've gone over in A&P, got all the things stuck in the membrane, things on the inside, things on the outside. And again, it does a lot of the same function. It's got some energy reactions. It's got regulating things in and out of the cell. It's selectively permeable for what it allows inside and outside of the cell. Big difference, though, for a cell membrane of a prokaryotic cell and a eukaryotic cell. In a prokaryotic cell, they don't have mitochondria. That's way too advanced. They're missing chloroplasts if it's a cell that can do photosynthesis. Way too advanced of an organelle. And so photosynthesis or ATP still has to get made somewhere in the cell. The cell still needs ATP. So in a prokaryotic cell, ATP is actually made out on the cell membrane because they don't have mitochondria. And so that ATP is actually made in the cell membrane of a prokaryotic cell. So that's a big difference from something you've learned in A&P. And again, if it's a photosynthetic bacteria, which we don't talk a lot about those in this class, it could do photosynthesis on the outside of the cell. In a plant, they would do that in a chloroplast, but that's way too advanced. Now we've gotten inside of the cell, and it is still a cell, so it still has cytoplasm similar to eukaryotic cells' cytoplasm. And prokaryotes still also have DNA, but the big difference is what they look like and where they're found. Prokaryotic cells don't have a nucleus, way too advanced of an organelle. And so we find the DNA in what's known as the nucleoid region. It's just an area inside the cell where the DNA is hanging out. Now, the DNA is found in chromosomes, but what they look like is a little different. So our chromosomes, if I had you draw out a chromosome, you'd probably draw an X. Our eukaryotic chromosomes are linear, and they also are found in pairs. And there's usually lots of them. For us, 46 chromosomes or 23 pairs of linear chromosomes. Well, in a bacteria, They have one chromosome. It's all the DNA is in that one single chromosome, and it's in a big old circle. So they have one chromosome, single and circular. But they do also, not all of them, but some bacteria have kind of this extra bonus DNA, and they're called plasmids. It's still circular in structure, it's still DNA, but bacteria that are found in these little bonus plasmids of DNA, they're not essential for the bacteria to survive. However, it is DNA, which means it can have genes, and those genes can code for something. And sometimes the coding of the different genes can help the bacteria. Might not be necessary for survival, but it can help with survival. And a lot of times that extra bonus DNA in the plasmids, it could contain genes for antibiotic resistance. There could be a snippet of DNA in a plasmid that knows how to reject some type of antibiotic or how to break down an antibiotic. And so it's not normally needed to keep bacteria alive, but that sure is an extra bonus if you can code for antibiotic resistance. Bacteria still have ribosomes. They're a little smaller, but they do the exact same thing. Same function as a eukaryotic cell ribosome, and it's to make proteins. Bacteria can also have what are known as inclusions or granules. And I'm like, these are going to be kind of storage devices. I like to just think of them as they're kind of like the vacuoles. They can store things, whether it's food or some type of pigment, but they can store. Bacteria also have cytoskeletons. It helps maintain shape. It can also help when cells divide. Some bacteria can make what's known as an endospore. Not all bacteria can make endospores, thank goodness. But the two biggest genera of bacteria that we're going to talk about in this class that do is anything in the Clostridium or in the Bacillus genus. And I apologize, these should be italicized because they are genus names. Bacteria that can make endospores. It doesn't mean they always are in an endospore state. They don't always have an endospore. If they're growing and dividing and just they're all happy, we call them vegetative. They're active, they're growing. When they go into an endospore state, they go dormant because conditions aren't favorable. And I'm like, it might be too hot, too cold, whatever it is. They can go into this dormant endospore state and they can stay in that endospore state for a very long time, sometimes thousands of years. And so when that bacteria realizes that it's in an environment that's not great for it, again, the temperature might be wrong, there might be chemicals along, maybe radiation, it's dry and they need water. They go into that formation of endospores. This is the whole process. I'm not all that, care that you need it. The big thing is that when it realizes conditions aren't favorable again, it doubles its DNA, so it's got fresh DNA, and then it makes a super extra tough protective coat. And it just hangs out like that. It's not reproducing, but it can withstand heat, drying, freezing, radiation, chemicals. It's really hard to kill when it's in an endospore state. And then as soon as conditions become favorable again, it goes back into the vegetative cycle and it starts to grow. It's called germination. Just like plants germinate means they are growing. Endospores, they are metabolically inactive. They are not doing anything. They're just sitting there. And I'm like waiting for conditions to become favorable again. Now, there are debates on how long they can stay in an endospore state. Thousands of years for sure. Some say maybe millions of years. And it could depend on the bacteria itself. So they're hard to kill, which means they're resistant to ordinary cleaning methods. Your normal Lysol is not going to kill this bacteria. So anything in the Clostridium bacillus or the Sporosarcina, but we're going to talk specifically these bacteria. Anything in the Clostridium and Bacillus genus, these are bacteria that can make an endospore. They can go dormant. And so to get rid of them, if they end up in the hospital, well, how do you kill them? Lysol's not going to kill them because they can go dormant and wait till the Lysol's gone, and then they can start to grow again. They can be on a surface, which is dry, or there's UV light, or there's radiation, and they'll go into an endospore state, and then they'll wait till that radiation is gone. They'll wait for the chemicals are gone, and they'll start to grow again. It's hard to kill. Even boiling is not going to kill these things. 100 degrees Celsius is not warm enough to kill these things. They're resistant to, extreme temperatures. Now, what will kill them is if you can increase the temperature enough. You need to get it to at least 120 degrees Celsius under pressure for at least 20 minutes. It's called autoclaving. And so boiling does not sterilize. Autoclaving can, because sterilizing means we've gotten rid of everything. There are no bacteria present whatsoever. And boiling will kill a lot, but it won't kill everything because you could still have endospores. Now, an example of a bacteria that you may have some personal or professional knowledge of that makes an endospore. Everyone's like, oh, really? Yeah, I'm gonna go back again to this slide because it's anything in the Clostridium or Bacillus genus, because we talk about these. Well, the very common bacteria that you may have heard of, and if you haven't, you will, is C. diff. Well, guess what the C stands for? C. diff stands for Clostridium difficile. Now, I should note, Clostridium is getting renamed to Clostridioides. It's still an endospore-making bacteria. They just realized there's lots of bacteria in this group, and so they're subdividing them out. Still C. diff, but C. diff is a bacteria that can make endospores. So if you have a patient that has C. diff, you can't just use Lysol. You can't just use boiling. It's not going to kill it. These bacteria are hard to kill. They can survive on surfaces for years. So you can't just think, oh, well, the patient that had C. diff, well, we cleaned the room and it sat there. I sprayed it all down. And I'm like, and they've been gone for like a week. It'll be fine. It's not. C. diff is hard to kill. They require really strong chemicals. And yes, strong enough bleach solutions will work, as well as autoclaving. And we'll talk about other bacteria in that Clostridium genus this semester. C. diff is just one of them. And they are hard to kill organisms because of that endospore option they can make. The cell wall of the bacteria, again, gives bacteria its shape. And the shape of a bacteria, how they like to hang out, and even sizes of bacteria are specific to bacteria. So when you look under a microscope and you see a round bacteria, we just don't say it's round, we call it a coccus-shaped bacteria. Well, that means that bacteria is always going to be round. And I'm like, unless it's pleomorphic, they don't change shape. Bacteria have a specific shape to them. And so some of the different shape that bacteria can have, they can be round or spherical. They're called coccus-shaped. They can be bacillus or rod-shaped. Some bacteria, they're not perfectly round, but you definitely wouldn't call them like long rods. Like almost like little short, plump rods. I'm like, they're still technically a rod, but they're not long, but they're not perfectly round. We just smush these two words together and we call them a coccobacillus. It's still bacillus, but it's not long. It's very, very short. There's another type of bacillus that has a curved shape to it. I'm going to show some pictures coming up. We call it Vibrio. And then there's some spirillums as well. So just some pictures. Here's a coccus. This is the round-shaped bacteria. We have a rod-shaped bacteria. They're very long. We've got a Vibrio. It's a rod, but it has a curve to it. Sometimes they even look a little more curved where they look like a V. These are usually more found in watery environments. A spirillum, it's going to generally be very long and coiled. And a spirochete are longer, and they don't always have quite that curl or coil to it, but they're going to have that kind of wavy kink to it. A few bacteria can branch off. Generally, the ones we talk about this semester don't, but a few can. A lot of fungi like to branch. And then there is a group that are called pleomorphic. Usually these are the ones that don't have a thick cell wall or any kind of cell wall, and they can vary in their shape. And so you wouldn't call these rod, you wouldn't call them round. They can change shape a little bit. Sometimes they'll look shorter, sometimes they'll look longer, some will look fatter, some will look skinnier, etc. Now, other than the shape of a bacteria like to hang out together in specific arrangements. And so it's species specific. Some only are found by themselves. They don't like to be near anything. We call them singles. Some like to hang out in pairs. Like anytime you look at them underneath the microscope, they're always found in pairs. They're called diplo, or sometimes we just call them pairs. Some like to hang out specifically in groups of 4. Some like to hang out in clusters, but irregular. It's, it's any arrangement. You could have a huge cluster and then a small cluster. Some like to hang out in long chains, and I'll show some pictures coming up. And some like to hang out in small little cube shapes. The bacilli also can still like to hang out in pairs. They can hang out in long chains, and some like to stack up on top of each other. So they're not in a chain, but they stack up on top of each other, what's known as palisades. So again, some like to hang out in singles, some like to hang out in pairs, some like to hang out in long chains. Here's an example where they like to hang out in groups of 4 or in little cubes. Here's just some of the clusters. And when you gram stain these and you get gram-positive clusters, they look like little balls of grapes or little bunches of grapes. Now, size of bacteria. Bacteria are small. Again, this is microbiology, which means for us to see bacteria, it requires a microscope. So in lab, you're going to practice with some microscopes. Bacteria, because this is just showing some of the size. Here's a yeast cell. Here's one of our white blood cells, a red blood cell. Here's the size of a common bacteria called E. coli. There it is, little green thing next to a white blood cell. So white blood cells you can see underneath microscopes when you magnify them 400 times. And 400 times you still wouldn't be able to see much of this bacteria or this little Staphylococcus bacteria. And so we have to make them even bigger. So we magnify them. And in microbiology, we magnify our organisms 1,000 times bigger than they normally are, and they're still going to appear very, very tiny. Bacteria, because of their size, we measure them in what is known as micrometers. And the symbol for micro kind of looks like a backwards U here, where the Staphylococcus has got 1 micrometer. It kind of looks like a backwards U. A micro is 1/1,000,000th the size of a meter. So we're magnifying it 1,000 times, but they're still tiny. We still have a lot more that we can magnify them. Now, viruses are measured in nanometers, which is 1/1000,000th the size of a meter. And our microscopes, even when we magnify them 1,000 times, you're still not going to see anything. Definitely not going to see any type of structure. Maybe it would look like a little speck of dust, but you wouldn't know if it's a speck of dust or a virus. So bacteria are generally measured in micrometers, 1/1,000,000th the size of a meter, 1 × 10⁻⁶. And viruses are measured in nanometers, 1/1000,000th the size of a meter, and measured, it's like 1 × 10⁹, 9 zeros, the billionth. Now when we do group and classify bacteria, so how we're going to ID them. Some of the things we look for is we look at what do they look like underneath the microscope? What color are they after a gram stain? What shape are they? What is their arrangement? We can look at some bigger structures. We can look to see what the colonies look like on a plate. And so if we grow these on a plate, we can look to see, does it have a certain size as a colony? Does it have a certain color, a different shape? Is it translucent? I'm like, are there any other structure or any other descriptions we can use to describe the colony? We can do some physiology. We can look to see, does it react with certain types of sugars? Because some do, some don't. We could do some serological tests. These are just some different immune tests. And we can also look at their DNA. And I'm like to help ID out the bacteria. Now, Bergey's manual is going to take all the bacteria out there, all the prokaryotic cells, and it's going to group them into 2 different domains, the Archaea and the bacteria. Those are 2 domains that have prokaryotic cells. And then it's going to subdivide it into other subgroups and different phyla. And I'm like, in the book, you can look through all the different ones if you're interested. We're going to start talking about some of the bacteria later, and we'll kind of talk about some of the groups and phyla that they're in. So this is just showing there are 3 domains of life. There's the eukaryote domain, which is, yes, we're there in the animal kingdom of the eukaryotic domain. And then you have an archaea and a bacteria domain. We're going to focus mostly on the bacteria domain. Viruses, not found here because they're not technically considered living, so we don't put them anywhere in the tree of life. So again, we're just using different structures, different features to help ID them. Now, some terms we use when we have classified an organism. A species, these are cells that share similar patterns, similar traits. Now, normally when we talk about species, we say, well, these are organisms that can breed amongst themselves and produce viable offspring. Well, prokaryotic cells don't breed. They don't have sex with each other. We're going to talk about how they do reproduce, but it's not by having sex. And so species kind of gets that line of like, well, how do we call them a certain genus and species? When it doesn't meet the species name. And so we are still going to be looking at what are some of the similarities. A strain is when we take one single cell and then we can grow it up, which means everything from it came from that single parent cell. So of course, they're going to be very similar. And then a type, this is a subspecies. So you can have a species of bacteria, but it can still be genetically different from each other. An example, because we'll talk about this when we get to it later. E. coli. E. coli is E. coli. Or is it? You guys are Homo sapiens. You are all Homo sapiens. You are all humans. You are all humans. You are all the same genus and species. However, are you all genetically identical? No. You are all the same genus and species, but you are still genetically different from each other. Bacteria are like that as well. And I'm like, there's more than one kind of E. coli. And I'm like, we call them different types of serotypes, because you'll see serotype come up in a future PowerPoint, because there are different genetic strains of the same genus and species. Just like you guys are genetically different, but same genus and species, so are bacteria. That's why not all E. coli are bad. They get a bad rap. Most E. coli are good. But apparently the E. coli, the certain serotype that, does cause lots of issues. Well, that's why we don't like E. coli. Now, some prokaryotes with unusual characteristics, we don't talk about these after this PowerPoint. These are just ones that are unique. And I'm like, these are ones that kind of grow or behave more like algae. They can create various types of sulfur and they live bad environments, or they even can do what are known as gliding. I'm like, they're just other bacteria that are out there. So cyanobacteria, these are photosynthetic bacteria. They don't cause human disease, so we don't really care about them. I mean, we do, but we don't. They're the sulfur bacteria. They're photosynthetic also. They don't give off oxygen as part of their photosynthesis, though. Instead, it's more of the sulfur smelly. Again, they're living, they're out there. They don't cause human disease, though. Don't really care about them. Then there's also some of these gram-negative bacteria that kind of ooze around as they grow. Also, don't cause human disease, don't totally care about them all that much. And anything that's in the Archaea domain, again, we really don't care about them. I mean, we care, they're living, so we care. However, Archaea bacteria grow in the really extreme environments. They love to grow in boiling water, which most bacteria die. They could care less. So some of the Archaea grow in boiling water. Some grow in the Dead Sea. It's not dead. There's bacteria. Some grow under, extreme acid, under extreme pressures at the bottom of the ocean. Like, they grow in very unique, harsh environments. Well, our bodies are none of those. And so nothing in the Archaea domain grows inside of us. So they don't cause us human disease. We don't really care about them. So that's a lot about cells, specifically prokaryotic cells in this chapter.